welcome to week number 5. This week what we will be focusing on is blood collection and hematology. So that means we are focusing on chapters 4 and chapters 5. Now, we did talk about chapter 4 last week in that video, so I'm only going to touch on the topic that will be relative to what we're learning in lab. And I'm really going to focus on chapter 5. So chapter 4, remember, is talking about blood collection. So last week we learned about the Vacutainer method. This week we're going to focus on the butterfly method. So if you did not complete the Vacutainer method successfully in week 4, you will be repracticing that this week for your grade. Now, for those of you who did successfully get the Vacutainer method, we'll be moving on to butterfly. The reason I don't let you move on is because you can't complete the second until you complete the first, okay? So butterfly method is really used when we have patients with smaller veins or have frail veins. For example, those can be veins found in our geriatric patients, our children, and those found in the hand. We really like to use the butterfly method with our children because it allows them to still have the range to move their arm while we still collect our sample. Where if we used a Vacutainer method, that's not easy for them to do, and they don't like to sit well for blood draws anyway. So the equipment that you'll need when using a butterfly method is a very unique and special design. First thing that you'll notice is that the needle is actually positioned between two plastic pieces that we call the wings. These wings are really used to help guide the needle into the vein, but also to hold the needle steady while you are collecting your sample. There is a tubing that comes off of the needle that goes into the patient's arm and travels down to where the tube will be placed onto the second needle. That tubing actually does allow a lot of freedom in the moving of the patient's arms and the tubing system itself. So that tubing can also be placed onto a syringe or the tubes for Vacutainer. Because of what we have in class, we will be learning how to do this with the tube method, so please come prepared for that, and that means that you'll also have to use a hub with that sheathed needle. When you're completing this procedure, there are a couple of things to keep in mind that can cause some problems. First and foremost, when you're drawing from a hand using a butterfly needle, you can cause a lot of pain. Nerve endings and actual nerves themselves are very close to the surface in your hand. So when we actually draw with that butterfly needle, we have the potential to hit those. So I want you to be very cautious when you're drawing blood from a hand with a butterfly needle. This procedure also is a little bit more painful, and people think, well, why is that? The needle's smaller. Just because the needle's smaller doesn't mean it takes the pain away. So this procedure does cause a little bit more pain than that of a Vacutainer. When you're drawing with a butterfly method, the order of draw that you used for Vacutainer is exactly the same as for the butterfly. I am not going to review that with you guys. Hopefully you're starting to get that under your belt from last week, not only when I talked about it in the video, but when we did the in-class assignment in regards to that. Alright, so that's chapter 4 for you guys. Let's start talking about chapter 5. Now chapter 5 was dealing with the study of hematology. And when we talk about the study of hematology, that's really looking at the visible elements or components in our bloodstream, and in our bone marrow. Hematology will also look at the process called homeostasis, which is really your body's ability to initiate a clotting response to stop bleeding, but also to help your body prevent getting blood clots in the future. It's its way of maintaining itself. The most commonly drawn test in a hematology department and for hematology studies will be a CBC, or a complete blood count. This is a test where you will examine the white blood cells, the red blood cells, platelet counts, hemoglobin, hematocrit, and differentials. Now, when dealing with hematology studies, there are some CLIA-waived tests that medical assistants can perform either in a lab or in a doctor's office. There are two specifically that I want to talk about for this week. One is a hemoglobin. The second is a hematocrit. Hemoglobin, like I stated earlier, is really part of your normal CBC. But sometimes doctors only want the hemoglobin result, not the whole CBC. So this test will look at the red blood cells by hemolyzing them or destroying them and measuring that against the released amount of hemoglobin in the blood. When you perform this test, the value that you will receive is generally read in grams per deciliter, or G per DL. This means that it measures the weight, or the grams, of the red blood cells against the volume of them. Now, when you do complete this exam, they will be different based on the type of patient that you have. The values you receive will be different. For men, they will have a result somewhere between 13 to 18 grams per deciliter. Women's will be lower with a range from 11 to 16 grams per deciliter. Children will have a range of 10 to 14 grams per deciliter, and newborns will be the highest from 16 to 23 grams per deciliter. You will complete a hemoglobin using a machine called a HemaCube, which is what you'll get to use this week in the lab. Hematocrits, on the other hand, are also part of your CBC, but we can do those individually as well. This test will measure the amount of packed red blood cells against the volume of packed cells, and then complete a total volume measure. This means that we will have to centrifuge the blood down and spin it down in order to get that test. When you perform this procedure, you will need to use two capillary tubes. Once both have been filled and then centrifuged, you can compare them against each other and take an average of the results that you receive. This result, unlike the hemoglobin, will be documented as a percentage, where hemoglobin is documented as grams per deciliter. But just like when we talked about getting the results for a hemoglobin, these values for hematocrits will also vary depending on the type of patient that you have. Men will have a range from anywhere of 42 to 52%. Women will have a range from 36 to 48%. Children will have a range from 34 to 42%. Infants will have a range from 32 to 38%. And as always, newborns will be the most concentrated, 51 to 60%. Alright guys, so those are the big topics that I wanted to touch on for this week's video. I also want to let you know that because we are going to be doing some more finger sticks this week, you should expect to get somewhere between 3 to 4 finger sticks this week. Those two other procedures that we'll be doing are both capillary procedures, so I want you to be aware that before you come into class. So please make sure you drink plenty of water and you've eaten before you come into class that day. Also, please read your chapters in full. I want to make sure that you are getting the information, and any questions that you have, you can bring to lab class. As a reminder for this week, your diversity rough draft project will be due Sunday by midnight. Remember, that should be a combination of the initial scenario you submitted to me, the feedback you received from myself and your classmates, and how you're really going to handle that situation. The rough draft is worth 60 points, so please make sure that you not only complete this assignment, but you complete it on time, because you don't want to lose that 10% for submitting it late. Also, this is one of those required assignments that I talked about in the beginning of the in the beginning of the quarter. You cannot move forward in your program if you do not complete this assignment, so please keep that in mind as well. Well, and also you can't complete a final draft until you complete a rough draft. Lastly, for this video, I wanted to remind you that your discussion post, your initial post is due on Tuesday, with your 2 responses due on Saturday. Any questions you guys have, please make sure that you bring them either to lab class or send me out an email or give me a call. I look forward to seeing everyone on Thursday. Have a wonderful week, guys. Bye-bye.